Good afternoon once again. I'm back still focusing on the C4 photosynthetic pathway. What I did in the first clip is to describe the process. How does it happen in the leaf? Why is it called a C4 pathway? That I think I did to the best of my ability. Now, the main focus is why did this pathway develop in plants? How does it benefit the plants? This kind of a pathway was discovered in plants that originated in areas that are very hot areas with very high temperatures. And as when we're dealing with photorespiration, we have tried to explain, the higher the temperature, the higher the photorespiration, because under high temperature conditions, oxygen even dissolves faster than carbon dioxide. Therefore, the combination of oxygen with RUBP, which will lead into photorespiration, it's even high at higher temperatures, which actually means plants finding themselves living in very high temperatures will suffer more from photorespiration, and they had to develop a mechanism to reduce or to minimize photorespiration. Now, let's not forget that photorespiration is about oxygen combining with RBP instead of carbon dioxide. And this is made possible because normally the air that gets into the leaf contains more oxygen than carbon dioxide. So basically what it means is, because RBP cannot discriminate between oxygen and carbon dioxide, if there is more carbon dioxide, then there will be more photosynthesis. But if there is more oxygen, there will be more photorespiration and not photosynthesis. So it means you can minimize photorespiration either by increasing carbon dioxide or by reducing oxygen. You increase carbon dioxide to be on the same level with oxygen or even above oxygen, or you reduce oxygen to be at levels lower than those of carbon dioxide. Now, this is how these plants minimize photorespiration. Carbon dioxide comes into the leaf, and within the mesophyll cells, that carbon dioxide is converted into bicarbonate. The bicarbonate has got a charge, a negative charge. So unlike carbon dioxide, which does not have a charge, the PEP carboxylase enzyme will never make a mistake within the mesophyll cells and allow oxygen to attach to PEP. Only the carbonate that has got a charge will be the one that attach to PEP, forming this organic acid. So these organic acids will be formed, and within them, remember, there is carbon dioxide hidden. So the next step is going to be these malate molecules that are formed within the mesophyll cells are delivered into the vascular bundle sheath cells, and they are delivered through an active process. They are pumped in high numbers into the vascular bundle sheath cells, into the chloroplast of the vascular bundle sheath cells. And once they are pumped in there, they are broken down. So the higher the number of malic acids or malate molecules pumped into the vascular bundle sheath cells, when each and every one of them is broken down, they are going to release CO2. Each time malate is broken down, CO2 is released. So you can imagine, with lots and lots and lots of malate molecules pumped into the vascular bundle sheath cells, and each and every one of these malate molecules being broken down, you're going to have so much CO2 molecules released from these malate molecules. Now, the oxygen would have entered the leaf. You can see it's entering into the leaf. There is the oxygen entering the leaf. Here within the mesophyll cells, that oxygen cannot play any role because this enzyme will never make a mistake of fixing oxygen. So that oxygen must find its way through the intercellular spaces up all the way until here, where there is a Rubisco enzyme. And the movement of that oxygen to the vascular bundle cells is going to be through a diffusion process, which is a passive mode of transport, which is not as effective as the active mode of transport through which these malate molecules are delivered to the vascular bundle cells and broken down into carbon dioxide molecules. So the end result here is within the vascular bundle sheath cells, where Rubisco is situated and which cannot discriminate between oxygen and carbon dioxide, there is going to be so much CO2 molecules that are coming from the broken malate molecules. This area is going to be flooded with so much carbon dioxide, and there's going to be very little oxygen. And therefore, because there will be so much carbon dioxide in this area where the Calvin cycle is supposed to take place and where RBP carboxylase, which cannot discriminate between oxygen and carbon dioxide, is located, there will be so much CO2, and this enzyme will have no choice but to fix carbon dioxide more instead of oxygen, because in any case, at this area, there will be so much carbon dioxide as compared to oxygen. What these plants do in the atmosphere, there is more oxygen, less carbon dioxide, but in these plants, at the point where carbon dioxide is supposed to be fixed to form the sugars, at the point where carbon dioxide is supposed to be fixed through the Calvin cycle that results in the formation of sugars, the plant makes sure that there is more carbon dioxide at that point, at that location, as opposed to oxygen. And therefore, there's going to be more photosynthesis taking place here than photorespiration. And in that manner, the C4 plant manages to suppress the process of photorespiration. They don't get rid of it completely, but they suppress it down to very negligible level. They push it down to lower levels to a point where this photorespiration cannot affect the productivity of this plant. It cannot suppress photosynthesis, therefore meaning it cannot suppress the growth of this plant, which is a huge matter. Because if photorespiration was to be allowed to be a big factor in plants that are growing in high temperatures, which in any case favors photorespiration, photorespiration was going to be a huge problem for these plants, to a point whereby they could not easily survive. They will grow so slowly, sometimes even die. Therefore, these plants developed a strategy to minimize photorespiration, which made it possible for them to be able to survive in areas that are hot. That is why the maize plants are grown in summer when it is very hot. That's why the sugar cane is also grown in summer where the temperatures are high. You can see sugar cane is mostly grown in KZN. KZN is not a very cold area. We don't know the KZN for being cold. We know it for being an area that is hot, that is humid. Mpumalanga also, where some sugar cane is grown. We don't know Mpumalanga to be an area where there is a lot of cold. The temperatures are very low. No. Of course, it does get cold here and there, but it's known to be a hot area, and these sugarcane plants thrive under those conditions. Northwest, it's also very hot. It can be very hot. Here in Limpopo, the Zeerust area. No, Zeerust is in the northwest. Here in Limpopo, there's a Bediela area. The Zeerust in northwest. Those of you who did agriculture, those are areas that are known to produce a lot of maize plants. These are C4 plants, and these plants can survive in very hot conditions. Even Thohoyandou area, you grow maize during summer. You know, temperatures in summer can be very hot, and these plants can survive in this kind of areas because they've got a strategy of minimizing photorespiration. On the other hand, C3 plants cannot survive that well in hot areas because they don't have a mechanism of minimizing photorespiration. That is why they grow best in areas that are cool, where the temperatures are very low, because under low temperature conditions, photorespiration is not a huge problem. Therefore, they can survive there, but they can't survive in very high temperature conditions. That is the advantage that comes with the process of photorespiration. Thank you.